So first, I'd like to thank the AAAS and the Lemelson Foundation for this unique opportunity to participate in this program, and also to thank my scientific mentors, Mark Davis and Chetan Khosla, who really have guided and supported me all these years. So I'm here as an Invention ambassador, but what I really want to tell you is that everything I'll talk about today and everything, in fact, that we do is actually a collaboration and a whole team of people. And so this is my group at the University of Washington. We are a group that works on developing biomaterials for different medical applications. And because we work at the interface of different disciplines, we are inherently a collaborative group. We have people in our group who are chemical engineers, molecular biologists, polymer chemists, and bioengineers. So for many years, we've been developing technologies for cancer treatment and for diseases of the central nervous system. But today I'm going to tell you a story that started just a few years ago that got us into a completely new area. And this story started with a meeting. A new faculty member, Nathan White, came to see me. Nathan is an emergency medicine physician, and he came to me with a problem and also a vision. Now, his problem was that he was losing patients who would bleed too much before they could reach the emergency room. And his vision was that we would be able to develop a technology that could help control bleeding and sustain life during this critical time point. Now, a little bit about trauma. I actually had not thought much about trauma until Nathan came to see me. But it turns out that trauma is the #1 killer of young people in the United States. It is the #1 killer for people less than 46 years old. And in fact, in this age group, trauma accounts for almost 50% of deaths. And this includes automobile accidents, acts of violence, falls, injuries, etc. And of this group of people, of victims of trauma, about a third of them have uncontrolled bleeding. They die because of uncontrolled bleeding. And it's generally believed in this trauma field that this point of uncontrolled bleeding is the most accessible way and the place where technology can really make an impact on saving lives. And so we were faced with this problem of how can we stop bleeding very quickly in trauma patients. And so Nathan and I sat together that day, and we taught each other about what we do, and we started brainstorming about a technology that we could use to give to patients immediately, help to stop the bleeding, and make them last until they can reach emergency care. And we came up with a list of design parameters of something that we would need to make this work. And it turns out that our 12 years of working in the drug delivery field actually held the answer to almost all of these design parameters. We wanted something that we could produce in mass quantities so it could be eventually used by many, and we knew how to do that. We wanted something that was biocompatible, that we could inject into the victims, that could circulate and help find the injury sites. And all our years of drug delivery, we developed the knowledge of how to do that. We wanted something that could act really quickly, but then it would leave the body within an hour, that critical time period that it takes to get a victim and transport them to a medical facility. And we knew how to design materials for that. We wanted something that could find clots and make them stronger by cross-linking them, by fortifying them. And we knew how to do that as well. And we needed something that would circulate, but then recognize the clot and see where the injury was. And that was our missing component. That was our problem. How could we have a technology that would recognize the clot? Now from here, I could see 3 possible routes. The first is that we could look in the literature and hoped that someone had found something that we could use. The second was that we would have to do what we usually do, which is design something from scratch. And the third is to do a library screening approach. And this I would best describe to you as panning for gold. We're looking through billions of different possibilities, trying to find those nuggets that have the properties that we need. Now let me set this up for you a little bit. This panning for gold, for example, I had a very talented student who did this panning for gold strategy. She worked on this for 3 years, over 10 different experiments, beautifully designed, but we were unlucky. And at the end of 3 years, we didn't find what we needed. And so this strategy, it can be successful, but at the same time, it can be a black hole, and it can suck in your time, your energy, your resources, and really your morale. So I recruited a very talented, very fearless grad student to come join us on this project, Leslie Chan. And I told her about this project. She was excited about it. And I told her about these 3 avenues that she could start to investigate. And I sent her off, as graduate students do, to start exploring the unknown. And to let you know, I mean, this was an unfunded project. This was something that we were doing out of our own interest. And we were piecing together, Nathan and I were piecing together bits of money we had here and there to make this work. And really, a year would have been a luxury for us in terms of money to seed into this project. And so my fear was that at the end of this year, we would still be trying to pan for this gold. But in fact, we sent Leslie out, and within a week, she came back to me with this email that just said, "How about this?" And she had found this paper that was just published 4 months prior by Peter Caravan at Mass General Hospital. And he had actually reported this exact missing piece that we were looking for. It was just amazing. So he had, in his research, been developing materials to bind to clots so that he could image them. And this was just what we needed. It had all the 4 S's that we wanted. It was specific, it recognized clots. It was stable. He had optimized this already so that it could be used in humans. It was species independent, which meant that we could use it for rats, but then have relative confidence that we could use the same thing for humans. And it was safe. He even tested this in clinical trials and showed that it was safe and well tolerated. And so Leslie, armed with this history of drug delivery that we had, these experiences in polymers, and then plus now this missing piece, was able to go right into the lab and get working. And she synthesized a polymer. And the very first polymer that she synthesized actually worked. And so in 3 months, she started this in July. In October, she was already able to show that this material at least strengthened and fortified clots in the test tube. And so with this, we started expanding our collaboration. We started working with Lilo Pazzo. She was the first collaborator to come and join us. She actually helped to characterize these hybrid clots made from natural materials and our polymer. We call our polymer Polystat because it's a polymeric hemostat. And this image here shows how we believe Polystat works. So the blue here is the Polystat polymer, and the multicolor is our natural fibrin, which is our matrix that forms our clots. And what Polystat does is it recognizes these clots, it goes in and it fortifies them, and it prevents them from breaking down quickly. So with Lilo, we were able to show that the structure of clots actually change once we put in Polystat, and it makes the clots stronger. From there, we added another collaborator, Xu Wang, an excellent surgeon. And Leslie and Xu developed an animal model of trauma that we could use to test Polystat. This is a lethal model. And so the animals that have this trauma don't survive. But amazingly, we showed in the first experiment again that all of the animals, 100% of the animals treated with Polystat are able to survive. And so this was our amazing outcome in these short 2 years. Now, as you can see, we are still in the very early chapters of this story. We're able to save lives, but right now we're saving rat lives. And what we really want to do is to translate this so that we can save human lives. And so what is our vision and where do we see this going? Well, I want to share with you, this is a report that came out recently. It studied the 10-year history of mortalities of US military soldiers. And what this report showed was that 90% of the potentially survivable mortalities were due to blood loss. And if you break this down further, you'll see that only 13% of these were due to traumas that happened in the limbs. The major dominance of victims were trauma due to inaccessible areas. Now, why is this? It turns out that this invention called the tourniquet has significantly reduced the mortalities associated with limbs. But we still don't have a technology that can be easily given in the battlefield or at the first scene of an injury to stop internal bleeding. And so our great hope is that Polystat might be an injectable tourniquet, something that the first responders, combat medics, or emergency medical technicians can inject into the victims on site, help to stop their bleeding, sustain them so they can be transported to a medical facility. And so this is our story, and these are the people that are involved. In fact, I am here as an ambassador for this group of collaborators that we've worked with. And so I've told you here about Nathan, who came to us with this problem and this vision that got us started in this whole area just 3 years ago. I've talked to you about Peter, who's our ghost collaborator. He doesn't even know that I'm talking about him today. He doesn't even know we exist. We have Leslie. She's the glue that really held us together and made this project work. Lilo, who helped us characterize the materials, and then of course Shu, who developed these animal models for the surgery. And so I hope that by listening to our stories, I would suggest to you that in the future when you think about inventors and inventions, that you realize that in most of these cases, it's not just one person, but it's a whole team of collaborators making these technologies available. Thank you for your time.